Today's Dr. Will's CBMD PhD. Today is Tuesday, September 20th, 2016, 12:09 p.m. Eastern Standard Time. The small G protein family. This includes the Ras superfamily, a group of smaller 21 kD monomeric GTP binding proteins anchored to the cytoplasmic surface of the cell membrane. 3 Ras proteins, HRAS, KIRAS, and NRAS, as well as a number of closely related proteins, RHO, RAC, are expressed in most mammalian cells. Signals from a variety of activated cell surface receptors include induce, RAS activation, RAS binding to cytosolic GTP is catalyzed by GTP-GDP exchange factors, cytoplasmic SOS protein. RAS binding to cytosolic GTP is catalyzed by GTP-GDP exchange factors, e.g. the cytoplasmic SOS protein. An activated RTK recruits an adapter protein GRB2 through its linker SH2 domains. SH2 ARC protein homology sites can serve as a recognition and coupling domains for tyrosine phosphorylation. Signaling proteins. Because GRB2 through its SH3 domains is constitutively associated with SOS, the RTK-GRB2 interaction brings SOS to the cell membrane in proximity to S, RAS, RAS, setting and motion into a process similar to that for a trimeric G protein. SOS increases dissociation rate of GDP by several orders of magnitude. GDP leaves RAS and replaced by free cytosolic GTP. RAS GTP is the active complex capable of inducing a cascade of intracellular phosphorylation events that result in cell activation. Proliferation and differentiation. RAS GDP is returned to its inactive state by GDPase activity, which splits off differentiation. RAS GDP is returned to its inactive state by GDPase activity, which splits off phosphate to reform RAS GDP. Because RAS hydrolyzes GDP slowly, returning off this system requires the participation of GDPase activating proteins. Gaps to accelerate the conversion of GTP to GDP. The RAS proteins function as molecular switches cycling between the active GTP-based on-state and the inactive GDP off-state. The propagation signal from RAS travels through both RAS-associated protein kinases and lipid kinases. One final common pathway is via the MAP kinases, mitogen-activated protein kinases. The sequence of activation is MAP-KKK to MAP-KK and finally to MAP-K. Activation of these 3 kinases require phosphorylation of both tyrosine and threonine sites. MAP-K then phosphorylates transcription factors, e.g. MYC, G, JUN, FOS that induce gene expression. The centrality of RAS signaling is shown. Stimulation of cell growth by RAS occurs G1 phase of the cell cycle. The RAS gene can be altered by a number of oncogene mutations. Many of these tumor-associated RAS mutants are resistant to the GTPase amplification by GAP. RAS therefore remains in its active GTP conformation, i.e. a turned-on signal resulting in deregulated cell stimulation, proliferation, and transformation. Protein signaling modules. Receptor-induced recruitment of protein relays the activation message along specific signaling pathways. Activated receptors or their targets interact with adapter proteins that contain specific domains or modules. These non-catalytic modules serve as intermediate linkers in the transmission chain. By recruiting specific proteins, adapters organize the signaling pathways into macromolecular complexes that determine the specificity of signal transduction. They recruit enzymes and bring them into close proximity with their substrates. One example is the SH2-SRC homology 2 module that is present in at least 100 adapter proteins. SH2 binds to phosphorylated tyrosine sites. The specificity of the linkage is dictated by SH2 recognition of a particular amino acid sequence adjacent to the phosphorylated tyrosine. Similar SH3 domains also function as linkers between specific proteins. Signaling pathways involve large numbers of proteins that contain these or other linker modules. The module will connect one protein to another, determining the specificity of the interactions necessary for a precise signaling route. Second messengers are rapid propagation of signals activated by the diffusion of small molecules released into the cytoplasm. These include CAMP, CGMP, IP3, and DAG. The release of these second messengers follows the activation of specific enzymes, e.g. adenyl cyclase, phosphorylase, NC, and nitric oxide synthase. Adenyl cyclase AC, this membrane-bound enzyme, when activated by a G protein, produces a molecular rearrangement of ATP with the loss of two phosphate groups to form cyclic AMP, CAMP, and CAMP to turn activates protein kinase A, pKa. CAMP serves as a second messenger for a number of hormonal signals in multiple target tissues. In the platelet, it is an inhibitor of aggregation. The signal is turned off when CAMP is degraded to 5-AMP by phosphophosphatases. Phosphatidyldecanol, a minor component of cell membrane, can be phosphorylated at multiple free hydroxyls on the inositol ring. Phosphoinositides, the phosphorylated derivatives of phosphatidyldecanol, are precursors of second messengers that regulate a variety of intracellular processes: proliferation, survival, cytoskeletal organization, and vessel trafficking. Phosphatidyldecanol, a biphosphate PIP2, is the major substrate for two enzymes, phospholipase CPLC and phosphoinositide kinase P13K. PLC, activated by stimulated G protein coupled receptors GPCRs, cleaves membrane PIP2, generating two second messengers, inositol triphosphate IP3 and diacylglycerol DAG. IP3 mobilizes calcium from this endoplasmic reticulum, which then associates with the protein calmodulin. The calcium-2 calmodulin complex binds to and alters the conformation of a variety of intracellular proteins and activates a series of protein kinases. Phosphoinositide 3 kinase P13K. P13 kinases are activated by ligand-bound growth factor receptors GPCRs and RTKs, and integrins. Activated P13K phosphorylate inositolipids at the E3 position of the inositol ring to generate the 3-phosphoinositide. The major substrate for P13K is phosphatidylinositol 4,5-phosphate. PIP2, the product is phosphatidylinositol 3,4,5-phosphate. PIP3 levels of these 3-phosphoinositides are very low in quiescent cells but rapidly increase upon activation of P13K. These lipids function in signal transduction by interacting with a variety of proteins containing 3-phosphoinositide binding motifs and recruiting the target proteins to cell membranes. The protein domains that recognize the 3-phosphoinositide include the plextrin homology pH domains and FYV domains. pH domains are found in a wide range of guanine nucleotide exchange factors for small g proteins, RAC, RHO, and ARF. Proteins containing the FYVE domain have been implicated in a variety of vascular trafficking events. Maintenance of 3-phosphoinositide at a low level is a primary function of the inositol phosphate PTEN. Dephosphorylation of these phospholipids switches off the signal. PTEN is the product of the PTEN tumor suppressor gene. Nitric oxide synthase NOS, 3 isoenzymes of this hemoprotein have been identified. NNOS, constitutively expressed in normal cells. INOS, typically expressed in inflammatory macrophages, and ENOS, constitutively expressed in endothelial cells. Nitric oxide NO, synthesized by NOS from L-arginine, is a diffusible intracellular messenger with multiple physiological functions, including a critical role in regulating blood pressure. NO diffuses out of the endothelial cell and enters the vessel smooth muscle cells, where it stimulates soluble guanyl cyclase to form CGMP. The increased levels of CGMP result in smooth muscle cytoskeletal changes and vascular dilation. GAG-diacylglycerol, GPCR, G protein coupled receptor, IP3 inositol triphosphate, PIP2 phosphatidylinositol biphosphate, PIP3 phosphatidylinositol triphosphate, PI3K phosphoinositide 3 kinase, PKC protein kinase C, PLC phospholipase C. Transcription factors. Transcription factors are signaling proteins that regulate gene expression. Some reside in an inactive form in the cytoplasm and on activation phosphorylation into the nucleus. Others reside within the nucleus. On activation, they bind to regulatory elements of DNA and turn on DNA transcription. They may bind to the promoter region in juxtaposition to the gene or to a more distant enhancer region. They form the link between the receipt of message and its translation into the cellular response. Transcription factors of primary interest in hematopoietic cells include STAT, signal transducer and activator of transcription, relays information from a number of cytokines. Cytokine binding to a cell surface receptor results in activation. Phosphorylation of JAK, which in turn recruits and phosphorylates tyrosine residues on STAT. Activated STAT dimerizes and moves to the nucleus where it regulates gene expression. This is the JAK STAT pathway. GAT1 regulates the expression of genes required for erythropoiesis, GATA. 1 recognizes conserved DNA, GATA, nucleotide motifs, and regulatory regions of a number of erythroid-expressed genes, including genes for heme synthetic enzymes, globin, and membrane proteins. NFKB nuclear factor KB is a cytoplasmic protein widely distributed in a variety of cell types. It is a transcription factor for a leukocyte adhesion molecule ICAM1, VCAM1, and E-selection, as well as for a number of cytokines and growth factors including GCSF and GM-CSF. In unstimulated cells, NFKB dimers are bound non-covalently to the inhibitor 1KB and associate which prevents nuclear translocation. Upon receipt of stimulatory signal, the complex dissociates, IKB is phosphorylated and degraded, and NFKB enters the nucleus. E2F is a transcription factor held within a pocket of the retinoblastoma RB protein. Upon phosphorylation of RB, E2F is released unbound. E2F activates expression of specific genes. Proteins encoded by these genes trigger the cell to move past the G restriction point and proceed into the S phase. Turning off the signal, the dynamic state of cell structure and function depends upon regulation of both protein synthesis and protein degradation. Temporal and spatial activation signaling pathways are regulated by mechanisms that modify or terminate the signal. Protein phosphatases may be divided into two groups based on substrate specificity. Tyrosine phosphatases specifically hydrolyze protein phosphotyrosine. They are important in the regulation of signal transduction pathways. The second group of phosphatases specifically hydrolyze serine threonine phosphosters that are essential to the regulation of cell cycle progression, DNA replication, and transcription. Intracellular proteolysis, the proteasome and ubiquitin are principal components of energy ATP-dependent proteolytic system that controls the intracellular concentration of many regulatory proteins. Proteins are targeted for degradation by covalent conjugation to ubiquitin, a 76 amino acid protein. Ubiquitin belongs to a family of cytoproteins. The attachment of ubiquitin to its target protein requires a sequential action of 3 proteins, an activating enzyme E1, a conjugating enzyme E2, and a protein ligase E3. E1 forms a thiester bond with a carboxyl terminus of ubiquitin in an ATP-dependent reaction. Activated ubiquitin is transferred to the cytosine in the active site of E2. E3 binding to both E2 ubiquitin and their target protein results in formation of an isopeptide bond between ubiquitin and the specific glycine residue on the protein. Successive ubiquitin molecules are linked through isopeptide bonds to form a polyubiquitinated chain. The polyubiquitinated provides the biological signal for protein degradation by the proteosome. The proteasome is a 26S cylindrical structure containing a catalytic core capped by 1 or 2 regulatory complexes. The proteolytic complex consists of multiple catalytic sites that degrade targeted proteins by ATP-dependent hydrolysis, releasing small peptides and free ubiquitin. Proteolytic degradation targets proteins engaged in processes that maintain cell function, differentiation, and mitosis. There is selective degradation of a variety of proteins, including components of the cell cycle, cyclins and inhibitors of CDKs, transcription factors, NFKB, and its inhibitor IKB. Protein oncogenes, degradation of cell signaling or the cell cycle is associated with oncogenesis. Proto-oncogenes encode a variety of proteins: the EGF receptor, the GTP binding protein, RAS, SRC protein, kinase, the growth factor, PDGF, and nuclear transcription factors MYC, J, UN, and FOS. Over 100 proto-oncogenes have been identified. Mutations usually result in dominant phenotype. Oncogenes are derived from their normal cell precursor, the proto-oncogene, by a number of mechanisms. 1-point mutations or deletions resulting in increased activity, e.g. RAS. 2-gene amplification with increased cell proliferation, e.g. cyclin D1. 3 translocation of fusion to produce a new protein with increased proliferative activity, e.g. BCR-ABL, in chronic myelolytic leukemia. Section 22. 4. Translocation of proliferative gene placed under the control of an enhancer gene, e.g. the translocation of MYC from a chromosome 18 to a vicinity of the immunoglobulin Ig heavy chain enhancer on chromosome 14 in Burkitt's lymphoma. The G proteins, mutations that alter G protein activation, result in a variety of diseases, including 1. The pituitary adenomous associated with acromegaly due to impaired GTP hydrolysis with prolonged transmission of signal, and 2. Pseudohypoparathyroidism characterized by resistance to a parathyroid hormone due to deficient signaling caused by genetic loss of the A-alpha subunit of G protein subtype GS. Vibrio cholera-toxin attaches to the G-alpha subunit, markedly slows GTP hydrolysis. The G protein is locked into its active GTP-bound form. The prolonged signaling results in increased cAMP synthesis with excessive loss of salt and water from intestinal mucosal cells. The RAS proteins acquired point mutations of RAS are associated with increased oncogenes. They are present in some 20% of patients with myelodysplastic syndrome and are associated with more rapid progression of the disease and shortened survival. Mutations are present in 20% to 30% of acute myelodysplastic leukemias. Point mutations are found in over 20% of cases of myeloma, chronic myelomonocytic leukemia, and large cell lymphomas. There is also a high incidence of mutations in solid tumors, up to 40% in colorectal tumors, and almost 100% in pancreatic carcinoma. The tumor suppressor gene PT and somatic deletions or mutations of this gene that governs dephosphorylation of 3-phosphoinositides are present in a large number of tumors, including glioblastomas, endometrial, and prostate cancers. The mutation frequency of PT and approximates that of P53. Germline mutations are present in Codin disease and autosomal dominant disorder associated with developmental defects and increased incidence of cancer. Good day, it's Dr. Will, CBMD PhD. It's Monday, September 19, 2016. It is 9:52 p.m. Eastern Standard Time. Cell signaling. Cell activation response depends on external signals and stimuli reaching the cell from its proximate environment or from a more distant source. Signals may arrive via the blood stream from specialized endocrine cells, hormonal stimuli from neighboring cells, paracrine stimuli, or from the cell itself, autocrine stimuli. The signal may be small hydrophobic molecules, steroid and thyroid hormones that transits the lipid membrane, binds directly to and activates its intercellular receptor, which in turn regulates the transcription of specific genes. Some signals are genes NO and CO that pass unhindered through the membrane to activate intercellular enzymes. Other signals are transmitted by direct cell-to-cell contact. However, many signals are large hydrophilic molecules that do not enter the cell but bind to their specific cell surface receptor and on docking activate the receptor and transduce a message to its cytoplasmic tail. Cell surface receptors are of 4 primary types. 1 ion channel linked receptors governing ion permeability, particularly important to synaptic neurotransmission and muscle contractility. They will not be considered here. G protein coupled receptors, 3 enzyme linked receptors, 4 integrins discussed in section 3. The signaling molecules are ligands for specific receptors that initiate cell response, manifest by activation, secretion, motion, cell cycle, progression, or other intracellular events. The transmitted signal is propagated within the cell by a series of mediators that include proteins and phospholipids, as well as small molecules such as cyclic, AMP, and calcium ions. Signal transduction by cell surface receptors. The signaling ligand docks on a specific receptor, which may be a superprotein 7-pass receptor linked to a trimeric G protein or a single-pass transmembrane receptor. The latter are of two types. One, those with intrinsic protein tyrosine kinase (PTK) activity. These are referred to as receptor tyrosine kinase (RTK). And two, those that lack intrinsic kinase activity but activate an associate PTK. This group includes the cytokine receptor. The ligand binding initiates signals that pass from the receptor to the nucleus via a cascade of phosphorylations mediated through a selected series of cytoplasmic proteins and phospholipids. Trimeric G proteins and their receptors. A G protein coupled receptor family with over 1,000 membranes is activated by diverse stimuli. Upon ligand binding, these cell surface receptors interact directly. Guanosine triphosphate GTP binding proteins, G protein, that serve as intermediates in the signal transduction chain. The G protein coupled receptor GPCR, G protein link receptors are all structurally similar. A signal polypeptide chain with an extracellular N terminus joined to the intercellular C terminus by a serpentine transmembrane region that weaves back and forth through the cell membrane 7 times. The ligand binding domain include the N-terminal segment and those segments of the transmembrane chain that protrude through the exterior surface of the membrane. The message is transmitted into the cell where the C-terminus and the intercellular portions of the transmembrane loops of the receptor interact with and activate the G protein located on the inner surface of the membrane. The trimeric G proteins, these proteins either stimulate or inhibit effector enzymes, thus altering the activity of the signaling mediators located on the inner plasma membrane in juxtaposition to the receptor. The heterotrimeric GTP binding proteins consist of non-covalently linked alpha, beta, and gamma subunits. The alpha unit is bound firmly to guanosine diphosphate GDP, but linked less tightly to beta and gamma units. These form a more closely associated beta-gamma dimer. In the basal unstimulated state, the opposition, the alpha subunit, is linked to GDP. On stimulation of the receptor, GDP is displaced by cytosolic GTP. The process is induced and accelerated by the activated receptor, functioning as a guanosine nucleotide exchange factor. The GTP alpha complex dissociates from the beta-gamma subunits. Both the GTP alpha complex and the abandoned beta-gamma dimer are capable of stimulating substrates. The system is now turned on. GTP and alpha may interact with multiple downstream effectors exposed on the inner surface of the plasma membrane, thereby initiating cell activation. In the basal unstimulated state, the opposition of the alpha subunit is linked to GDP. On stimulation of the receptor, GDP is displaced by cytosolic GTP. The process is induced and accelerated by the activated receptor, functioning as a guanosine nucleotide exchange factor. The GTP alpha complex dissociates from the beta-gamma subunits. Both the GDP alpha complex and the abandoned beta-gamma dimer are capable of stimulating substrates. The system is now turned on. GTP alpha may interact with multiple downstream effectors exposed on the inner surface of the plasma membrane, thereby initiating cell activation. The duration and intensity of the signal is dependent on and limited by the bound GDP. The intrinsic GTP ACE activity of the alpha subunit hydrolyzes the bound GTP and GDP, turning the signal off. This GTP ACE activity is enhanced by the binding RGS, regulator of G protein signaling, proteins to the alpha subunit. The RGS protein are GTP ACE activating proteins, GAPS, which function as negative regulators of the signal. Binding of these proteins to the GTP alpha subunit accelerates hydrolysis of the bound GTP to GDP alpha, then recombines with the beta-gamma complex with reconstitution of GDP alpha beta gamma. The system resumes its basal state. The activated G protein may 1. Activate protein kinase that phosphorylate tyrosine, serine, threonine, cytoplasmic proteins, rendering them active transmitters of message. 2. Interact with membrane-associated adenyl cyclase to synthesize cyclic AMP and cAMP. 3. Activate the phosphoin, cytide pathways. 4. Activate the RAS-MAP kinase pathways. The enzyme links cell surface receptors. These receptors have an external ligand binding domain, a signal past transmembrane segment, and cytoplasmic domain. Most of these receptors belong to the receptor tyrosine kinase RTK family, in which the cytoplasmic tail and intrinsic kinase activity. Kinases are the enzyme that add phosphate groups to selected amino acids in a protein. The docking of ligand induces receptor dimerization, the juxtaposition of two cytoplasmic cytolytic domains that allows transphosphorylation to the catalytic sites, followed by autophosphorylation of additional tyrosine residues and further activation of the receptor. This sets in motion a cascade of protein phosphorylation via a series of serine, threonine, and tyrosine kinases, resulting in cell activation. Many of these ligands are growth factors, e.g. epidermal growth factor, EGF, and platelet-derived growth factor, PDGF. The downstream phosphorylation of enzymes and structural proteins regulates the cell cycle, differentiation, shape, and adhesion. Receptors that lack intrinsic kinase activity are coupled to non-receptor protein tyrosine kinase. These include the cytokine receptors for erythropoietin on red blood cell precursors, interleukins on lymphoid cells, and GCSF and GMCSF on hematopoietic cells. The cytokines binding to these receptors produce dimerization and a conformational change that induces phosphorylation activation of associated protein tyrosine kinase, a member of the Janus family of PTK, JAK, 1, 2, or 3. The Roman deity Janus, guardian of the gate of heaven, is represented with two faces, one face directed outward, the other inwards to the temple. The aptly named JAK, PTK are intermediates in the transfer of signal. They first phosphorylate receptor subunits and activate a transcription pathway through phosphorylation of STAT, signal transducers and activators of transcription. Phosphorylated STATs dimerize and migrate to the nucleus as inducers and regulators of transcription of specific genes. The cytokine signal also articulates with the RAS and P13K pathways. Integrins. The integrins are cell surface receptors that interact with complementary adhesion molecules to integrate the external environment with the interior of the cell. They transmit signals from the extracellular environment to activate intracellular responses. Intracellular mediators. Signal transmission and amplification involves a network of proteins that transform external stimuli into an appropriate cellular response. Propagation may proceed by protein phosphorylation of tyrosine, serine, and threonine sites through a network of protein kinases. Termination of the signal depends on dephosphorylation by specific phosphatases or proteolysis.